Welcome back to the deep dive. Today, we are opening a file that feels a little bit like a cold case mystery, honestly. But it's one where the victim profile is heartbreakingly specific, and the perpetrator was hiding in plain sight for decades.
Yeah, it's a really fascinating topic. And for those of you who are joining us with your textbooks open, which we always highly encourage, we are diving deep into Murray's medical microbiology, specifically Chapter 51.
Right.
We're going to use that as our structural spine today. We will also be pulling some key historical and virological details from Jowett's Chapter 37, just to really round out the picture. But Murray is our main roadmap.
Perfect. So, Chapter 51, the reoviruses. Now, before we get into the molecular nuts and bolts, and I know you love the nuts and bolts, I want to set the scene properly for you. I want to give you a clinical vignette because medicine is ultimately about stories, right? Not just protein structures.
Oh, I agree completely. Context is everything. If you can't recognize the patient, it really doesn't matter if you know the genome.
Exactly. So picture this. It is mid-January. It's freezing outside. You are a third-year medical student or maybe a resident working in the pediatric emergency department. The waiting room is just full of coughing kids, but in walks a mother carrying a 6-month-old infant. And immediately your spidey sense goes off. This baby just looks wrong.
Okay, let's drill down on that. What are we seeing when you say wrong?
The baby is miserable, but also really lethargic. The mother tells you this started about 2 days ago. It began with vomiting projectile, frequent vomiting. Then came the fever, low grade, maybe 38 degrees Celsius. But then came the diarrhea. And we aren't talking about a loose stool here or there. We are talking about persistent, watery, explosive diarrhea.
Yeah, the kind that parents describe as totally uncontainable.
Precisely. And now you look at the child's physical signs. The eyes appear sunken into the orbits. The fontanelle, the soft spot on the top of the head, is depressed. You check the mouth, and the mucous membranes are tacky and dry. You pinch the skin on the abdomen for the skin turgor test, and instead of snapping back, it just tense. It just stays there.
Right, so medically speaking, we are looking at moderate to severe dehydration.
Yeah.
This is a hypovolemic state pending.
Exactly. This isn't just a simple tummy ache. This child is losing fluids way faster than they can take them in. The mother also mentions the baby goes to a daycare center 3 days a week. So here's the mystery. We have a winter setting, a 6-month-old watery diarrhea, vomiting, and rapid, profound dehydration. Who is the culprit?
Well, I mean, if you're a betting person and you see this specific presentation, the classic winter vomiting disease in a temperate climate like North America or Europe, you have to put your money on rotavirus.
Rotavirus, the absolute king of pediatric gastroenteritis.
It really is. It's the single most important cause of severe gastroenteritis in infants and young children worldwide, and it belongs to the family we were discussing today, which is Reoviridae. But before we get to the virus itself, we really should talk about that family name, because reovirus is actually a bit of a misnomer, or at least a historical curiosity.
I love a good etymology story. Break it down for us. REO.
So it stands for respiratory enteric orphan.
Orphan? Like it has no parents.
Nope, not quite. Orphan in the sense of a virus looking for a disease. When these viruses were first discovered back in the 1950s by giants in the field, like Albert Sabin, actually, scientists found them in the respiratory tract and the enteric tract of healthy people.
The gut, right.
The gut. They isolated the virus, but they couldn't pin any specific pathology on them. They were just present. So they called them orphan viruses.
That is incredibly ironic.
It is the ultimate irony, because this orphan turned out to be the parent of one of the most significant killers of children in history. As Jowett's points out, historically, before vaccination, rotavirus was responsible for up to 500,000 deaths annually in preschool children, mostly in developing countries.
500,000 children a year from an orphan virus that they thought did absolutely nothing.
Exactly. It just goes to show that in virology, you really shouldn't underestimate something just because you haven't found the smoking gun yet. And while we see less mortality in developed countries today due to better access to IV fluids, the morbidity is still just massive. It fills up hospitals every single winter.
Okay, so we've identified the culprit. We know the stakes. Now we need to understand the weapon. We need to look at the structure of this virus. And for everyone listening, if you want to ace your exams, whether it's the USMLE or your shelf exams or whatever, you need anchors. You need mental models. And for reoviruses, we have a perfect one.
We do. It's a structural rule that defines the entire biology of the virus.
I call it the double-double rule.
I like that a lot. Let's unpack the double-double. What's the first double?
Double number one is the genome. This virus has double-stranded RNA.
Which is huge. And we need to pause here, because if you just gloss over double-stranded RNA, You miss the entire biological drama that is happening inside the cell.
Why is that so dramatic, though? I mean, our DNA is double-stranded. Why can't RNA be double-stranded?
Because of the context of where it is. Think about a human cell. We keep our double-stranded DNA locked safely inside the nucleus. In the cytoplasm, where the machinery of the cell works, we have single-stranded mRNA. We do not typically have long strands of double-stranded RNA just floating around in our cytoplasm.
So if a cell sees double-stranded RNA in the cytoplasm, it knows something is deeply wrong.
Exactly. It is a massive red flag.
Yeah.
In immunology, we call this a PAMPA, a pathogen-associated molecular pattern. Our cells have sentries, specifically toll-like receptors, TLR3 to be precise, that are constantly scanning the cytoplasm and endosomes looking for dsRNA.
It's like a security guard looking for a specific type of weapon.
Right. And if they see it, they hit the panic button immediately, they trigger the interferon response. And this is a scorched earth policy. The cell shuts down protein synthesis, it tries to degrade the RNA, and it often just commits suicide apoptosis to stop the spread of the virus to neighboring cells.
So the virus is essentially walking into a trap. By its very nature, its genome is a beacon that says, I am here, come kill me.
It is. Which means the virus has to be incredibly clever to survive. It can't just let its genome float around naked in the cytoplasm like a poliovirus might with its single strand. It has to hide it.
Right.
And that brings us to your second double.
Right. Double number 2 is the capsid. It has a double-layered capsid.
Or even triple-layered in the case of rotavirus specifically. And this is critical.
So describe this for us. Is it like a thick wall?
Imagine a fortress within a fortress. Or a better analogy might be a Russian nesting doll. You have an outer shell, and inside that you have another protein shell, and inside that the core. Most viruses are happy with just one capsid. Reoviruses invest a huge amount of energy into building 2 or 3.
And this isn't just for show. This armor is highly functional. Why do they need such heavy armor?
Think about where this virus lives. The human gut. The gastrointestinal tract is arguably the most hostile environment in the entire human body for a pathogen.
It's an absolute war zone.
It is. First, you have the stomach, pH of roughly 2.0. That acid will denature most proteins instantly. Then if you somehow survive that, you get dumped into the duodenum where you were hit with bile salts.
And bile is a detergent, right?
Exactly. Its job is to dissolve fats so we can absorb them. If you are an enveloped virus like influenza or coronavirus, you have a lipid membrane, bile will just rip that membrane apart through saponification.
So the flu virus would die instantly in the gut.
Pretty much, yeah. That's why you don't get stomach flu from influenza. But reoviruses are non-enveloped. They don't have that lipid weakness. And their multi-layered protein capsid is incredibly stable. It can survive a pH range from 3.0 all the way up to 9.0. It laughs at lipid solvents like ether or chloroform. It is a tank.
So the double-double gives us the basic identity. Double-stranded RNA implies it's a massive immune target, and the double capsid implies it's an environmental tank built to protect that target.
Correct.
Yeah.
But there is a plot twist here involving those gut enzymes that is just absolutely fascinating.
Oh, I love a plot twist. Is this about the trypsin?
Yes. So we mentioned the gut is hostile. It's full of proteolytic enzymes like trypsin and chymotrypsin. Their whole job is to chop up dietary proteins so we can absorb the amino acids. Most viruses try to hide from these enzymes or they get destroyed by them. Rotavirus actually needs them.
Wait, it wants to be chopped up?
To a degree, yes. When rotavirus is released from a host in the stool, it's not actually fully infectious yet. It's essentially locked. When it enters a new host and hits the small intestine, the trypsin cleaves a specific protein on the outer capsid called VP4.
VP4, that's the spike protein.
Exactly. It stands for viral protein 4. When trypsin cleaves VP4, it transforms the virus from a mildly infectious particle to a highly infectious particle. It primes the spikes so they can act like grappling hooks to penetrate the next enterocyte.
That is devious. It's like saying, oh, you threw acid and enzymes at me. Thanks, I needed a haircut to get ready for the party.
That's essentially what happens. It turns the host's primary defense mechanism into an activation switch. This is exactly why rotavirus is so wildly successful in the gut. It evolved specifically to thrive in that soup of digestive enzymes.
Okay, so we have the double-double, double-stranded RNA and a double or triple capsid. But there is one more structural feature that is absolute exam gold. And for this, I want you to picture a deck of cards.
Yes. Segmentation. This is the third pillar of reovirus biology.
So the genome isn't one long, continuous string of genetic code like a scroll. It's chopped up. It's segmented.
Right. Reoviruses generally have 10 to 12 distinct segments of double-stranded RNA. For rotavirus specifically, it is exactly 11 segments. Each segment corresponds to a specific gene coding for 1 or 2 proteins.
And why does this matter practically? Why do we care if it's one piece or 11 pieces? Is it just a trivia fact for the boards?
No, it is evolutionarily critical. It matters because of what happens when two different strains of the virus infect the exact same cell. The shuffle. Imagine you have a blue deck of cards representing strain A and a red deck of cards representing strain B. If they both infect the same cell at the same time, the cell becomes a mixing bowl. All the segments of the cards get thrown onto the table. When the virus starts packaging new viral particles, it just grabs 11 segments. It might grab 10 blue cards and 1 red card.
And this is called reassortment?
Exactly, genetic reassortment. This is the exact same mechanism that makes influenza so dangerous and why we need new flu shots every single year. Rotaviruses can do the exact same thing.
So hypothetically, if a human rotavirus and, say, a bovine rotavirus from a cow infect the same cell, maybe in a farmer or a child living near livestock, they can swap segments.
Yes. And suddenly you have a hybrid virus. Maybe it has the outer coat of the cow virus, but the replication machinery of the human virus. The human immune system looks at it and says, I don't recognize this outer coat, and mounts no immediate defense.
And that leads to genetic shifts.
Precisely. It's a major driver of viral diversity and evolution. It allows the virus to jump species barriers and evade population immunity. But as we'll see later, it's also a tool we have harnessed to create life-saving vaccines.
We'll put a pin in that for the vaccine section. So structure check. Double-stranded RNA is the PMP, double capsid is the armor, and the segmented genome allows for shuffling. Now let's talk about the heist. How does this virus actually get into the cell and replicate without setting off those TLR3 alarms? We mentioned earlier that dsRNA is a red flag, so how does it hide?
This is the safe house strategy, and it connects directly back to that double capsid structure we just talked about.
Walk us through the invasion. The virus attaches.
Right, it attaches to the intestinal epithelial cell, the enterocyte, specifically near the tips of the villi. It uses that VP4 spike protein we mentioned that got primed by trypsin. It gets taken up into the cell via endocytosis. Now, usually this endosome fuses with the lysosome, which is the cell's recycling center full of more enzymes.
Usually that's where viruses go to die, right? The lysosome is the incinerator.
But remember, rotavirus likes enzymes. The acidic lysosomal environment triggers a structural change. The outer layer of the capsid is stripped away. But, and this is the absolute key, the inner capsid, the core, remains completely intact.
So it sheds its heavy winter coat but keeps its shirt on?
Exactly. This subviral particle, or core, is released into the cytoplasm, but the double-stranded RNA genome is still trapped safely inside it.
Wait, if the genome is stuck inside the core, how does it replicate? Doesn't the RNA need to come out to be read by the host cell ribosomes?
This is the genius of reoviruses. The core is a mobile factory. The virus brings its own enzyme, an RNA-dependent RNA polymerase, inside the core with it.
Because human cells don't have that enzyme at all.
Correct. Human cells can't make RNA from an RNA template. We go DNA to RNA. So the virus brings its own photocopier. It sits inside the closed core and transcribes positive-sense single-stranded mRNA from the double-stranded genomic template.
It's doing all the dirty work inside the safe house.
Yes. The blueprints, the dsRNA, never leave the safe. The factory just churns out single-stranded mRNA copies. These copies are then extruded through tiny pores at the vertices of the capsid-like ticket stubs coming out of a machine.
That is just brilliant engineering. So the single-stranded mRNA comes out into the cytoplasm, and the cell's sensors, the toll-like receptors, they don't mind single-stranded RNA. They just think it's normal cellular traffic.
Exactly. It completely flies under the radar. The host ribosomes grab this viral mRNA, translate it into viral proteins, and the infection takes hold. The virus has effectively bypassed the innate immune system by never letting its red flag genome touch the host cytoplasm.
And as these newly synthesized proteins build up, they congregate in these dense clusters in the cytoplasm, right?
Yes, these are called viral factories or inclusion bodies. If you look at an infected cell under an electron microscope, you can see these dark, dense blobs, often associated with microtubules near the nucleus. That's where the assembly line is running, putting new cores together.
There's one weird detail about rotavirus assembly that Murray mentions. It's a bit of a biological curiosity, but it feels like the kind of gotcha question you see on an exam. Something about a pseudo-envelope.
Ah, yes, the pseudo-envelope. This is a bit of trivia that loves to show up on advanced exams because it contradicts the fundamental rule we just established. We said reoviruses are non-enveloped.
Right. They are naked viruses.
But during the assembly process, the immature rotavirus particle actually buds into the endoplasmic reticulum, the ER. Now, when a virus buds through a host membrane, it usually keeps it as an envelope. So for a brief moment in time, The immature rotavirus actually has an envelope.
So it is enveloped.
Only for a minute. As it matures inside the ER, that transient membrane is stripped off and the outer capsid proteins lock into place. It's like trying on a coat, checking yourself in the mirror, and then deciding, I'm tougher without it, and throwing it away.
It's just showing off at this point. I can make a membrane and destroy it just because.
It creates a paradox. A non-enveloped virus that requires membrane budding for its assembly pathway. It's very unique.
Okay, so that's the biology, the tanks, the safe house, the factory. But we are here to talk about the impact on that 6-month-old baby in the ER. Let's talk about rotavirus specifically as a pathogen, the clinical star of the show.
Right. So let's quickly touch on classification. There are groups A through H.
Which one do we actually care about clinically?
Group A, Murray, makes this very clear. Group A is the major human pathogen responsible for the severe infant diarrhea we're discussing. There are others. Group B, for example, has caused massive outbreaks in adults in China, sometimes called the adult diarrhea rotavirus. But for your boards and for general pediatrics in the West, Group A is the main player.
And we classify them further using serotypes. You mentioned VP4 earlier.
Yes, the classification is binary based on the 2 surface proteins that the host immune system actually sees. VP7 is a glycoprotein, so serotypes based on it are called G-types.
And VP4?
VP4 is protein-sensitive. Remember the trypsin cleavage we discussed? So those are called P-types.
G-types and P-types. It sounds like a Myers-Briggs personality test. I'm a G1P8.
In a way it is, but it's incredibly important because neutralizing antibodies, the immune system's guided missiles, target these specific proteins. So when we make vaccines, we need to make sure we cover the common G and P types that are actually circulating in the wild. If the vaccine covers G1, but the local outbreak is driven by G2, it might not work as well.
Got it. Now let's get to the mechanism of the disease itself. I call this the lawnmower and poison model. It really helps me distinguish the 2 distinct types of diarrhea happening in this infection.
That's a really good distinction. Let's start with the lawnmower. This refers to the structural damage in the gut, right?
Yes. Explain what rotavirus does to the villi. We need to visualize the anatomy here to understand the physiology.
Okay. The small intestine is lined with millions of tiny finger-like projections called villi. Their main job is to vastly increase the surface area of the gut. Think of a deep shag carpet. That deep pile gives you a massive surface area to absorb water and nutrients compared to a flat floor.
And who lives on these villi?
The cells at the very tips of the fingers are the mature enterocytes. These are the highly specialized workers. They have the brush border enzymes like lactase to break down complex sugars, and they have the transporters like SGLT1 to absorb sodium in and water. They are the heavy lifters of the gut.
And rotavirus targets them specifically.
It does. It preferentially infects them, replicates inside them, and kills them. It effectively mows down the tall grass. This leads to what pathologists call villus blunting or villus atrophy.
So instead of a shag carpet, you suddenly have a flat, bald floor.
Exactly. And if you lose that surface area and you lose those mature specialized cells, you completely lose the ability to absorb sugars and electrolytes. The sugars, things like lactose from milk or sucrose, just stay in the gut lumen unabsorbed.
And this is basic physics at work. Water follows solute.
Osmosis. The high concentration of sugar sitting in the gut lumen pulls water out of the body tissues and into the intestine. That causes osmotic diarrhea. This is also why these kids often have very acidic stool. The normal bacteria in the gut ferment that undigested sugar into lactic acid and other organic acids.
That's the lawnmower, malabsorption. But that's not the whole story, is it? Because this diarrhea is explosive. It's profound. And that's where the poison comes in.
This is a critical point, and one that represents a major historical discovery in virology. Rotavirus makes a specific protein called NSP4, non-structural protein 4.
And why is NSP4 such a celebrity in microbiology?
Because for a very long time, we thought only bacteria had enterotoxins. Things like cholera toxin or E. coli heat labile toxin. Viruses were just supposed to kill cells directly. But NSP4 acts as a viral enterotoxin, the first one ever discovered.
So the virus literally produces a poison?
Essentially, yes. NSP4 is secreted from infected enterocytes. It then binds to specific receptors on neighboring uninfected cells in the crypts. It triggers a complex signal transduction pathway involving intracellular calcium. It causes the intestinal cells to release their intracellular calcium stores.
And what does that massive calcium surge do to the cell?
It tells the cell to dump everything. It causes the active secretion of chloride ions into the gut lumen through specific channels. And where chloride goes, sodium and water rapidly follow.
So it's a devastating double whammy.
Yes. You have the lawnmower preventing absorption, which gives you osmotic diarrhea, AD. You have the poison-forcing active secretion, which gives you secretory diarrhea.
Plus, there's evidence that NSP4 stimulates the enteric nervous system too, right?
Yes, it triggers the release of serotonin, or 5-HT, from enterochromaffin cells in the gut. This serotonin stimulates the vagus nerve, which directly activates the vomiting center in the brainstem.
So you have projectile vomiting, profound malabsorption, and active water secretion. No wonder these babies get dehydrated so incredibly fast. They're losing fluid from both ends, and their gut is actively pumping water out while refusing to absorb any back in.
Precisely. It's a perfect physiological storm.
Uh-huh.
And that leads us right back to the clinical presentation we talked about at the start, the triad.
Vomiting, fever, and watery diarrhea.
It usually starts with vomiting. That's often the very first sign parents notice. Then comes the low-grade fever. Then a day or so later, the profuse watery diarrhea hits.
And let's talk about the stool characteristics. I'm sorry if you're eating lunch while listening to this, but we have to go there for the clinical picture.
It's important for diagnosis. The stool is often described as rice water or just frankly watery. But here is the absolute key negative finding for your exams. There is usually NO blood and NO leukocytes, or white blood cells, in the stool.
Why is that absence so important?
Because it helps you instantly distinguish rotavirus from bacterial dysentery. If you have a patient with Shigella salmonella or Campylobacter, those bacteria invade the gut tissue deeply, causing intense inflammation, ulceration, and tissue destruction. You get gross blood and pus, which are fecal leukocytes. Rotavirus causes malabsorption and secretion, but it doesn't cause that deep, bloody, inflammatory mess.
High yield differentiation right there. No blood think viral or maybe cholera. Blood think bacterial invasion.
Generally, yes. That's a very solid rule of thumb for the boards.
Let's talk numbers and epidemiology. We said this is a winter disease.
The winter vomiting disease.
Yeah.
In temperate climates, like the U.S. or Europe, you see a very sharp peak in the cooler months, usually January through March. In the tropics, it's less seasonal. It can happen year-round, but it often peaks in the drier, cooler season.
And the age group. You mentioned 6 months to 2 years. Why not younger babies? Why not older kids?
Well, under 6 months, babies often still have some maternal antibodies transferred across the placenta, which is IgG, or through breast milk, which is secretory IgA. That gives them some passive protection. It's not total immunity, but it's usually enough to prevent severe life-threatening disease.
And older kids.
By age 3 or 4, almost every single child on Earth has been infected at least once.
They've earned their stripes in the daycare trenches.
They really have. They have developed natural immunity. They might get infected again because the immunity isn't perfect and there are different serotypes, but subsequent infections are usually much milder or even completely asymptomatic. The real danger zone is that specific window between losing mom's protection and gaining your own robust immunity. That's the 6 months to 2 years mark.
And the transmission route.
Fecal oral. And it is incredibly efficient. Remember that sturdy double capsid? It survives on toys, changing tables, doorknobs for days or even weeks.
And the infectious dose is tiny, right?
It's terrifyingly low. Some sources say as few as 10 to 100 viral particles can cause a full-blown infection.
And how much does a sick baby shed in their diaper?
An infected infant can excrete up to 100 billion particles per gram of stool.
That is a math problem that equals an absolute outbreak. 10 particles to get sick, billions upon billions coming out.
It is. That's exactly why it rips through daycare centers and pediatric wards like wildfire. Nosocomial infection, which means getting sick in the hospital, is very common with rotavirus. You admit a kid for pneumonia, and 3 days later they have severe rotavirus diarrhea because it's spread from the room next door via a nurse's hands or a shared stethoscope.
So how do we actually diagnose it in the hospital? Do we just look at the kid and say, yep, it's winter, you're puking, it's rota?
Clinically, often, yes, that's exactly what you do. But if you need to formally confirm it, you don't do a viral culture. It's historically very hard to grow in standard cell lines. You use antigen detection.
Like an ELISA or EIA test.
Right. You take a stool sample and look for the viral antigen, usually VP6, which is the major intercapsid protein. It's fast, it's sensitive, and it's cheap. PCR is technically the gold standard for pure sensitivity and genotyping strains. But the rapid antigen test is the everyday clinical workhorse.
And what about electron microscopy? I always see pictures of it looking like a little wheel in the textbooks.
That's actually where the name comes from. Rota is Latin for wheel. Under the EM, the double rim looks like the tire, and the structural proteins radiating out from the center look exactly like the spokes of a wheel. But nobody does EM for routine clinical diagnosis anymore. That's purely for research or classic textbook images.
Okay, let's go back to the ER and save the baby. Treatment.
Supportive care. There is no specific antiviral drug for rotavirus. And you definitely don't give antibiotics because it's a virus. You have to aggressively replace the lost fluids and electrolytes.
ORS, oral rehydration solution.
It is arguably one of the greatest medical advancements of the entire 20th century. It's a very specific, scientifically calculated mix of glucose and electrolytes.
Like glucose, though. Why not just give them salt water?
This relies on a specific transporter in the gut called SGLT1, the sodium-glucose-linked transporter. This transporter moves one molecule of glucose and one molecule of sodium together into the cell. Even in a gut severely damaged by rotavirus, this specific transporter often continues to function better than the others. By giving sugar with the salt, you trick the gut into absorbing the sodium, and water always follows sodium.
So the sugar is basically the Trojan horse that drags the water back into the body.
Exactly. It bypasses the damaged cyclic AMP and calcium pathways.
And if they are too sick to drink, or if the vomiting is just too severe to keep the ORS down,
then you have to move to IV fluids immediately,
right.
Isotonic saline or Ringer's lactate. The ultimate goal is just to keep the intravascular blood volume up until the gut heals itself. The cells lining the intestine turn over very rapidly, usually every 3 to 5 days. If you can keep the kid hydrated and alive for that 3 to 8 day window, the gut will repair itself. The villi will grow back and they will recover completely.
So the difference between life and death is really just keeping the tank full while the body fixes the leak. But prevention is always better than cure. Let's talk about the vaccine. And this is a story that needs some real time because it is a bit of a roller coaster. It's a story of incredible triumph, tragedy, and eventually redemption.
It really is. We have to go back to the late 1990s. The global medical community was absolutely desperate for a rotavirus vaccine. You had 500,000 kids dying every single year.
And they came up with RotaShield?
Yes. A live rhesus human reassortment vaccine. It was licensed in the U.S. in 1998. It was highly effective at preventing severe disease. Everyone celebrated.
But the celebration was very short-lived.
Very short. Within about a year, reports started trickling in to the VAERS system, the Vaccine Adverse Event Reporting System. Pediatricians were seeing a slight but noticeable uptick in a very specific condition in babies who had just been vaccinated.
Intussusception.
Yes. Intussusception.
That is quite a mouthful. Explain what that actually is physically happening in the body.
It's a telescoping of the bowel. Imagine a telescope collapsing in on itself. One segment of the intestine, usually the ileum, slides inside the adjacent segment, the cecum.
Like a snake swallowing its own tail.
Sort of, yeah. And when that happens, it drags the mesentery and the blood vessels right along with it. The blood supply gets compressed and cut off. The tissue swells. It causes a mechanical blockage. And if not fixed, eventually necrosis or tissue death. It is a major surgical emergency.
And RotaShield was causing this.
The epidemiological data showed a statistically significant association. It was rare, maybe 1 in 10,000 vaccinated infants, but the risk was undeniably real. Because the mortality from wild-type rotavirus in the U.S. is very low since we have good hospitals and IV fluids, the risk of the vaccine was deemed completely unacceptable. It was voluntarily withdrawn from the market in 1999.
That must have been a devastating blow to the researchers and public health officials.
It was catastrophic. It set the whole field back years. And it left the developing world where kids actually were dying of rotavirus every day without any vaccine at all. There's a huge ethical debate in the medical community. Was it right to pull a vaccine that could potentially save 500,000 lives globally because of a rare side effect that was mostly manageable with surgery in developed nations?
That's an incredibly heavy question. But eventually science bounced back.
It did. Fast forward to 2006, we got 2 brand new vaccines, RotaTeq and Rotarix.
And these were tested more rigorously than almost any vaccine in history, specifically because of that RotaShield shadow.
Exactly. The clinical trials for these were massive. They involved roughly 60,000 to 70,000 infants each. They had to definitively prove safety regarding intussusception before the FDA would even look at them.
So let's meet the new recruits, RotaTeq.
RotaTeq is a pentavalent vaccine. That means it covers 5 different common strains. It is a human bovine reassortment.
Oh, they used the shuffle trick we talked about earlier.
They did. They took a bovine rotavirus, which naturally doesn't make humans sick, and they swapped in the surface proteins, the gene P types, from human strains. So the infant's immune system learns to fight the human surface proteins, but the backbone of the virus itself is weak and attenuated in humans.
And the other one, Rotarix.
Rotarix is monovalent. It's just one single strain. But it's a fully human strain that has been heavily attenuated or weakened in the lab, so it could replicate a little bit but not cause actual disease. It provides very broad cross-protection against multiple serotypes.
Now, both of these are live vaccines, and both are given orally.
Correct. They are drops placed directly in the mouth, no needles involved.
Why orally, though? Why not a standard intramuscular shot like the polio or hepatitis B vaccine?
Because we specifically want mucosal immunity. If you inject a vaccine into the muscle, you predominantly get IgG antibodies in the bloodstream. That's great for preventing a virus from spreading through the blood, which is viremia. But rotavirus isn't a blood virus, it's strictly a gut surface virus. We need antibodies waiting right there in the gut lumen.
We need secretory IgA.
Exactly. By giving the vaccine orally, we stimulate the gut-associated lymphoid tissue, or GALT, to produce huge amounts of IgA that gets secreted right into the intestine. We're putting the guards directly at the gate where the invader will arrive.
Makes perfect sense. And are these new ones safe? What about the telescoping bowel issue?
They're considered very, very safe. The post-licensure data over millions of doses suggests there might still be a tiny increased risk of intussusception, maybe 1 to 5 cases per 100,000 infants vaccinated. But the benefit of preventing severe rotavirus gastroenteritis just overwhelmingly outweighs that microscopic risk. The clinical impact has been staggering. Hospitalizations for rotavirus in the U.S. dropped by over 80% almost immediately after these vaccines became routine.
That is a massive public health victory. But because of that history, there are a couple of strict contraindications you have to know for the boards, right?
Yes. 2 major absolute contraindications. First, a history of intussusception. If a baby has had intussusception before, for any reason, you do not give the rotavirus vaccine. It might trigger recurrence. And second, SCID, or severe combined immunodeficiency. Because it is a live virus, it can replicate unchecked in a baby with no functioning immune system and cause severe chronic life-threatening diarrhea.
Got it. So history of intussusception or SCID equals no rotavaccine.
Correct.
Okay, before we wrap up the reovirus file, there's one other cousin in the family we need to mention. It's not a gut issue, and it's not worldwide, but it is a reovirus, and it shows up on exams.
Right, you're talking about Colorado tick fever.
Yeah.
Caused by a cultivirus.
This sounds like a classic travel medicine question. Where do we find this?
Well, the name pretty much gives it away. The mountainous western United States. The Rockies, usually at altitudes above 4,000 feet. It's transmitted by the wood tick, Dermacentor andersoni.
So the classic vignette is someone hiking in Colorado in the spring or summer who gets a tick bite.
Right.
And what are the symptoms?
It looks a lot like dengue fever or really bad flu. Sudden onset of fever, chills, severe headache, muscle pain or myalgia, and photophobia, which is light sensitivity. Sometimes they get a rash. But it often has a classic saddleback fever pattern. That means fever for 2 to 3 days, then a break where they feel better for 2 days, then the fever returns again.
And what is the key differentiator for the exam? How do I distinguish this from Rocky Mountain spotted fever, which is also in that area?
Well, RMSF is caused by rickettsia, a bacteria, and typically has a petechial rash that specifically starts on the wrists and ankles and spreads inward. Colorado tick fever is viral. But the specific biological quirk of cultivirus is that it targets and infects erythroid precursors in the bone marrow.
The stem cells that make red blood cells?
Yes. The virus actually matures inside the red blood cells and just stays there. Since red blood cells live for about 120 days and don't have a nucleus to mount an immune response, the virus is completely protected from the host immune system inside the cell. You can find viral antigen hiding inside RBCs for weeks or even months after infection. That is a very unique feature.
So if the board question mentions a hiker in the Rockies with a biphasic saddleback fever and leukopenia and virus inside red blood cells, you immediately think Colorado tick fever.
Exactly.
All right. We have covered a huge amount of ground today, from the orphan that wasn't to the double structure, the lawnmower and poison pathogenesis, and the whole vaccine roller coaster.
It's a dense chapter for sure, but hopefully the mechanisms make intuitive sense now rather than just being a list of facts to memorize.
Let's summarize. What are the absolute big takeaways you want the listener to hold on to before we hit the quiz?
Okay, let's boil it right down. First, identity. Reoviruses stands for respiratory enteric orphan. Second, structure. Remember the double-stranded RNA, which acts as a PMP and triggers TLR3, and a double or triple capsid that acts as an environmentally stable tank against stomach acid and bile. Third, genetics. It has a segmented genome, usually 11 segments for rotavirus. This allows for reassortment, the card shuffle, and genetic shifts. Fourth, clinical. Rotavirus is the leading cause of severe diarrhea in infants aged 6 months to 2 years with a strong winter seasonality. Fifth, pathogenesis. It causes malabsorption through villi blunting the lawnmower and secretory diarrhea via the viral enterotoxin NSP4, the poison. And finally, prevention. We use live oral vaccines, Rototec and Rotarix, to stimulate mucosal IgA, and you must watch out for a history of intussusception.
And the ultimate, so what? Why does all of this matter to you as a future doctor?
Rotavirus is a perfect testament to the power of molecular understanding. We identified a mass killer. We understood its structure, that tough double capsid, perfectly explaining its insane environmental transmission. We discovered the first ever viral enterotoxin NSP4, which literally rewrote the microbiology textbooks. And we used its own genetic trick reassortment to create a vaccine that has saved countless lives. It's a massive victory for modern medicine, but it's one we have to constantly maintain with ongoing vaccination programs.
Well said. Okay, listeners, don't take your earbuds out just yet. It is time for the rapid recall. This is where we actively cement the knowledge we just built. I'm going to throw some single best answer style questions at you. I'll pause after the question and I want you to answer it loud. Scare your roommates or the people on the bus. It's totally fine. And then the expert will give you the correct answer and the why behind it.
Let's do it.
Question 1. A 1-year-old infant is brought to the ER in February with severe dehydration following 3 days of watery diarrhea and vomiting. Stool analysis reveals a non-enveloped virus with a segmented double-stranded RNA genome. Which specific viral protein is functioning as an enterotoxin to cause fluid secretion in this patient? Think about the poison.
The answer is NSP4, or non-structural protein 4. Why? Because NSP4 acts as a viral enterotoxin, triggering calcium release from the endoplasmic reticulum and subsequent massive chloride secretion into the gut lumen. This adds a profound secretory component to the osmotic diarrhea already caused by malabsorption. For review, see Murray Chapter 51, the pathogenesis section.
Question 2. During the replication cycle of reoviruses, the virus transcribes its mRNA inside the viral core before releasing it into the cytoplasm. Why is this containment strategy absolutely necessary for viral survival? Remember the immune sentries.
To prevent the host cell from detecting double-stranded RNA. Why? Because dsRNA is a very potent PMP recognized by toll-like receptors, specifically TLR3 and other cytoplasmic sensors. If it were exposed, it would trigger a massive interferon response and cellular apoptosis. The virus hides the dsRNA inside the core to evade innate immunity. See Murray Chapter 51, the replication section.
Question 3. A medical student asks why rotavirus is transmitted so efficiently in daycare centers and is so difficult to eradicate with standard cleaning. Which structural feature of the variant confers this stability against environmental stressors, stomach acid and bile? Think about the armor.
The non-enveloped multi-layered double or triple capsid. Why? Because unlike enveloped viruses, which are easily destroyed by bile and drying out the dense protein layers of reovirus, are highly stable across a very wide pH range and are totally resistant to detergents and desiccation. See Murray Chapter 51, the structure section.
Question 4. A new strain of rotavirus is identified that contains genetic segments from both human and bovine rotavirus strains. What specific genetic mechanism accounts for this mixing of genomic material? Think of the deck of cards.
Reassortment. Why? Because the genome is segmented co-infection of a single enterocyte by 2 different strains, allows the RNA segments to be randomly shuffled during packaging, creating a completely new mosaic strain. This leads to major antigenic shift. See Murray Chapter 51, the genetics section.
Question 5. A 4-month-old infant is scheduled for their first dose of the rotavirus vaccine. The mother mentions that the baby's older brother had a telescoping bowel problem when he was an infant. While the brother's history isn't a direct contraindication, if this baby had a personal history of that condition, the vaccine would be strictly contraindicated. What is that condition officially called? The surgical emergency.
Intussusception. Why? A personal history of intussusception is a strict contraindication for rotavirus vaccination due to the very small potential risk of recurrence or vaccine-associated intussusception. SCID is the other major absolute contraindication. See Murray Chapter 51, the prevention section.
Question 6. A hiker returning from the Rocky Mountains presents with fever chills and photophobia. A peripheral blood smear shows viral antigen hiding within mature red blood cells. Which virus is the most likely cause? Think of the cousin.
Colorado tick fever virus, which is a cultivirus. Why? This is a reovirus transmitted by the wood tick in the western U.S. The absolute hallmark of this infection is the targeting of erythroid precursors, leading to the prolonged presence of the virus protected within mature red blood cells. See Murray Chapter 51, the other Reoviruses section.
And there you have it. You are now officially a reovirus expert.
You are. So go forth, wash your hands thoroughly, and always respect the double-double.
I want to leave you with one final provocative thought to mull over. We talked about how rotavirus uses trypsin, a host defense mechanism, to become more infectious. What other viruses might be out there quietly turning our own physiological weapons into activation switches? It makes you look at host pathogen interactions in a completely different light, doesn't it? Something to think about. See you in the next deep dive.